So this is the fourth video in a series on factor analysis by Dr. Jennifer Ripley at Regent University. And this is factor analysis step by step. So this is kind of walking through how you actually conduct a factor analysis, and I'll try to explain concepts as we go through, but really the purpose of this video is how do you actually conduct one. So the first thing to check in all statistics is their assumptions. Factor analysis doesn't have too many assumptions, and they're all pretty familiar except for maybe one new one. So screening for missing data, as always. Determine if variables are continuous, good ones, or categorical. If you have categorical variables that you want to put into a factor analysis, they really should be dummy coded, basically put into a very small continuum, because that's one of the assumptions for factor analysis. If you have good continuous variables, look for outliers. So typically in my training that I do, I have people just run a quick Mahalanobis distance to see if they have outliers. This is a bigger deal if you have very large variance. So if maybe people could answer questions on a scale of 1 to 100. If people can only answer questions on a scale of 1 to 5 or something like that, it's really hard to get an outlier when your scale is only 1 to 5. But you can run a quick one and see if you have any outliers going on. You can look at a scatter plot to see if you have linearity of items. Most people just kind of look at a correlation matrix and make sure that things correlate with each other, at least moderately. Again, you're trying to find like that 0.2 to 0.8. It's not a problem if it's too low or too high. You're probably just going to be throwing out some items if they're too low especially. Normality, so just looking at skewness. Again, skewness by item might be important if you have items that have like a 1 to 100 scale on them that you included in your study. If they're very small and they're just a 1 to 5 or something, just check and make sure you don't have like a really high skewness, which would mean essentially that you don't have, like everyone's answering 4 and 5 essentially on their answers, and nobody answered 1, 2, 3. If you have a very small scale. So, and that's the case, you might just have kind of a bad item, and you could, use normality as a reason to throw the item out even before you start your factor analysis. There's something called sphericity, which is a KMO statistic, and sampling adequacy. Your textbook talks a little bit about this and how, what it looks like in SPSS. I'm not going to go into it in detail because people argue whether or not it's even necessary to do this. So, but just be aware that they're there. Collection of data. Okay. Oops, sorry. Just keeping you awake here. All right. The next thing I want to show you is what a Scree plot looks like. So this is a different set of data. And this one, if we were using Kaiser's rule there on the bottom, you can see that we would only have one component because only the first component has an eigenvalue of greater than 1. Components 2 through 5, they're less than 1. So this we would say is, oh, we have a single component of study, single factor. If it was a factor analysis. So Scree plot here, we look at it. This is actually just taking those eigenvalues and putting them on a plot. So you'll see like 2.78 is the first one. It's actually that first dot there is at 2.78, and the second one is at 8, and then 0.51, and it goes down like that. So what people will sometimes do is they'll look at the 1, but they'll also look at the Scree plot, and they'll see like, where did the data start to drop off? And it's kind of like, well, kind of looks like maybe at 2 in this one, right? And then you kind of see it level off at that point. You could argue 1 because all the data is really in 1, but you could also argue 2. So Scree plot is kind of like looking at scatter plots. It's not like super obvious, but it can be helpful to help you kind of understand what your data looks like by looking at it plotted instead of just looking at the numbers. Similar to like, what is the distance between each? All right. Then you have to make a decision standard. So usually you pick the closest one, which is the highest factor score, and then at least maybe a 0.3 difference, or loading a certain level, such as a 0.3, maybe both. When we look at it, it might make more sense. For my labs and exams, that sort of thing, I will tell you what the rule is. So, if it doesn't fit that standard, then you kick the item out of the study. It's not going to stay in your scale, and you're going to kick it on out. So this is a picture. What we have here is a scale that I developed about marriage and career and priorities kind of thing, like willingness to sacrifice type stuff. And so you have each of the questions there on the left. There's 5 questions. okay? Then you have factor 1 and factor 2. So we decided that we have 2 factors in this study. And what we want to do is to see for these 5 items, do they fit better with factor 1, so line 1, or factor 2? So which island do they fit on better? To go back to our island metaphor. For this, it's pretty obvious for some of them. You can pretty quickly tell, like, okay, the first question, the higher number is 0.25, so it fits better with factor 2. The second question is factor 1, then factor 2, then factor 2, then factor 2. Right? Okay, so all of them are in factor 2 except for the second question. But the issue is we've got a couple of funny ones here along the way. So, for instance, that last one, a spouse's first priority should be achieve personal goals through his or her marriage. It falls on factor 2 at a 0.309, but it falls on factor 1 at a 0.174. That's actually not that different. Like if you subtract those out, that's only, what, a 0.13-ish? So that's not a very big difference. So a rule that you might pick is, okay, it needs to fall at least 0.30. Okay, it does that for that last question, but it's not 0.2 or 0.3 different. So you might either pick 0.2 or 0.3 different, meaning it's distinguished onto one and it's far from the other, or decently far. In this, the first question would also go if that was our rule of 0.30, because while it falls on factor 2 at 0.25, that's not at least 0.30. What that means is that first question isn't really close to either line. It's pretty far away from both lines. It's very far from line 1 at 0.03, but it's pretty far still from line 2 at 0.25. So this is a pretty simple process. When you do this in the actual work, you just go through and kind of circle which ones, and you have a rule, and then you just go through with your calculator and subtract. Sometimes it's obvious, that it's more than a 0.3 difference or whatever, but other times it's not obvious, and you have to actually pull your calculator out to make sure that it didn't make it along the way. Okay, there's another thing that I want you to understand, and that is something called rotation. When SPSS runs a factor analysis, it can leave the factors kind of as they naturally fall, like their natural spot that they fall in, but it can also do something called being free to rotate, and it's most common to actually rotate the data because there's nothing magical about the lines falling in this kind of like perfectly up and down, right to left spot. They really could fall anywhere in the data and it's fine. So you have to tell SPSS, all right, let it rotate, let it turn and fall anywhere in the data, and do it by using the maximum variance possible, which is the whole point of factor analysis, which is why people tend to use Varimax. And so it'll move the factors and rotate it. So it looks kind of like this. Like in the first one is where they naturally fall, but it's kind of a forced spot, and it doesn't really even make sense that it would be there. It makes more sense that you would tell the computer, put the lines in the best spot possible to maximize variance, because that's kind of the whole point of factor analysis anyway. So just to be aware of what rotation is, almost every single article I've ever read is a Varimax rotation in psychology, so it's common. There are a couple of limitations to PCA that you want to be aware of. One is that there's a lot of art to the data. You can get different results with different sets of data. It's a lot of what people call like taking the data, throwing it up on the wall and see what sticks. So if you read an article and they've done a factor analysis on a scale, but it's the only time it's ever been done, it's still pretty preliminary at that point, right? So we think there's probably a factor being measured behind the scenes and everything, but we don't know. So it's not until you get to a confirmatory factor analysis that you have some reputation on that measure. Now, a lot of times in dissertations and stuff, you have to pick a measure and it's just like the best you've got. And they did a factor analysis on it and it's only been done once, and that's just the best measure you have. But just know that your chances of having problems with that measure are higher. If you have problems with a measure, you have less chance of finding things in your dissertation to be significant because you have more error. So just be aware, the better the measure, the better the study. In PCA-FA and definitely CFA, you need pretty good sample sizes. So 200 at least is considered a fair. 300 is really what people like to see. And then you need normality and linearity, and you don't want outliers. They can cause problems. And you want factorability. So your correlations should be about 0.3. If you're finding you have hardly any correlations, falling at a 0.3, and that when you look at your initial correlation matrix, then you probably aren't going to find anything anyway. You can run it, but you're probably going to find just a mess of data. It's not going to factor well. All right, so big picture. Deal with the assumptions, right? And you got a list here, and your textbook also has a list on your principal components analysis or factor analysis. Answer the questions. How many factors should there be? Is it 1, 2, 3, 4? Typically Kaiser's rule, that whole eigenvalue of one thing is typical, but you look at other things too. What kind of rotation should I do? Typically a Varimax rotation. And then how much variance do my factors account for? Each of them. So each line, how much variance does it account for? And then once you've done that and you said, okay, I've got 2 factors, it's accounting for 75% of the variance or whatever. Now for each individual item, which factor is it associated with? So which island is it on? And which items need to be disregarded and thrown out? They shouldn't be used in future research. How good are the factors in terms of being distinguishable? Are they really something different, or are they really similar to each other? And then you can run an internal consistency test to see if your individual factors or subscales are consistent, or if you might have a problem. A final term to understand in your Grimm and Untitled chapter, and this will pop up in multiple regression 2, which is all about lines. They'll talk about residual. Residual is another word for error. So I just wanted to throw that in there and give you this picture. This is a picture of residuals. So if you have dots around a line, whether it's a regression line in this case, or whether it's an eigenvector, the distance of every dot from the line is considered a residual. Because if it was perfect, they'd all be on the line, but it never is. You always have some error. So the distance between where the dots actually are and the line is considered the residual. Important thing to know.